Today's lesson is going to introduce Unit 5, and it actually is talking about Lesson 5.1, and we're also going to talk about Lesson 5.2 and get into Lesson 5.3. So we're going to actually tackle a lot in the next 10 minutes. So just taking a look, I pointed out to students that this is Chapter 5 in the textbook. This is an excellent chapter for you to read before you come to class or after you come to class, just to kind of reinforce the concepts. Genetics is the study of inheritance, and inheritance is the process of passing traits from parents to offspring. So if we take a look at Gregor Mendel and we look at his work was done in 1866, and his work wasn't actually discovered until the 1900s. So in his lifetime, he really didn't, he probably knew the value of it, but it wasn't appreciated. Remember, they didn't have Twitter. So at the time he was doing his research, there was a theory of inheritance called the blending theory, and it was that offspring are a blend or mix of their parents. And Mendel kind of disagreed with this because he didn't really notice that there was a blend. For example, if tall pea plants were crossed with short pea plants, he didn't see medium-sized pea plants that would be the blending theory of inheritance. Now, there is some exceptions to what the rules are, but this he disproved this with some of his experiments. He chose pea plants. They were fast, they grew fast, they were easy to raise, just like scientists today work with things like fruit flies, Drosophila melanogaster, because they're cheap, they're easy to grow, and they follow the model of his principles very well. So he had to tackle the whole experimental design and controlling his experiment. And so plants typically self-pollinate, which means they have male parts and female parts. And if allowed, the pollen from the anther goes to the stigma or the pistil where the female gametes are, and that plant self-pollinates. Mendel chose to use cross-pollination to make sure that he could cross, and he didn't know they were genes at the time, cross information from one plant to the other. So to do this, to control this, he did cut off the male parts of one plant, the female parts of the other, and then he used a paintbrush to transfer the pollen. So some basic information to have is the P generation is the parental generation. F1 stands for first filial, and F2 stands for second filial. So these are referencing generation 1, generation 2, generation 3. So he crossed a tall pea plant with a short pea plant, and what his outcome was, all tall plants. He no longer saw the short pea plants. But when he took two plants from the F1 generation and he crossed those two plants from that generation, he got a ratio of 3 tall plants for every 1 short plant. So that kind of reappeared. So this is how he worked to make some of his principles. And before we get there, let's just talk about we're focusing on genes that are inherited. So it's a trait that's inherited from your parents. There are other types of traits that are acquired. So it's something that's learned, practiced, or caused by the environment. And there's a practice question here that kind of explains this. If you have questions, please see me. But going back to inherited traits, that means that it would be passed on the chromosome, and areas of the chromosome that have instructions for that particular trait are called genes. So these are the 7 traits that Mendel studied. Now, for example, the gene for stem length had 2 different versions. It had a long and a short, or I call it a tall and a short. So what the term allele is it's another name for the version of the gene. So this one had a long allele and this one had a short allele. What he found was that some appeared to dominate over others. So he would call them the dominant allele and a recessive allele. And as he went along, he identified the dominant, the recessive, the dominant and the recessive for all of these 7 traits. He kind of lucked out because it's not this simple. But using that information, he kind of started to realize that this is passed on. And now we know with the chromosomal theory of inheritance, we understand that we get a chromosome from our mom and a chromosome from our dad. And when those come together during fertilization, we have now a homologous pair. So On, let's say, chromosome number 1, there is a location, let's say it's down here at the end, where you'll find the gene for flower color. And maybe mom's chromosome is containing instructions for purple flowers, an allele for purple flowers. But dad's allele contains instructions for white flowers. So each cell has 2 copies of the chromosome and that form the homologous pair. So you're always going to have 2 copies. Down here at the bottom, we drew chromosomes where parents had the same allele for white color, or this set of parents had the same allele for purple colors. So we use terminology. This is hybrid, this is homozygous, and homozygous. Again, pointing out reading 5.4 is very helpful. So here's Mendel's principles, and they're not laws because there are exceptions to it, and reading more about this in 5.2 is helpful. So the first one is the principle of dominance. One allele is dominant over the other. The second, principle of segregation, which sperm or egg is chosen, like which basically the alleles that original cell has when it's going to become a sperm, those chromosomes are going to separate to become haploid. So which one gets which information? And then independent assortment is like, how do chromosomes line up in meiosis during metaphase 1 of meiosis? And are they inherited together, or are they on the same chromosome, or are they going to separate? So we'll take a look at these principles as we go. Probability is a math term that most of you are familiar with, and brush up on it by reading 5.5. It's the likelihood or chance that an event will occur. So we need to use this language carefully. If we talk about observed, it's what actually happens. If we talk about expected, it's what should happen. So here's some more information about the rule of multiplication and addition. We rarely use addition. So focus on just kind of reading over about multiplication, and we'll use it in the next week. Phenotype refers to what an organism looks like. So it's physical traits. And I underlined pH, so you can kind of match that vocab word up. It's what it looks like. So we would use words like yellow, red, white, descriptions. Genotype is an organism's genetic makeup. And we use a capital letter or a lowercase letter to represent what gene or what allele that organism has. The term homozygous is referring to a zygote that has the same alleles. So here we have homozygous dominant would be big R, or homozygous recessive would be little r. If they have the opposite, we would say it's heterozygous, or hybrid carriers is another name for heterozygous. They have opposite alleles. They have 2 different alleles. So setting up a Punnett square is pretty simple. You draw a box. This is going to represent a monohybrid cross. So we're just going to look at one trait. We're going to put the parents' gametes on the outside. So this is the mom. So her possible eggs would contain a little t and a little t. And this is the dad. His sperm could be either big T or a little t. Next, you fill in what the possible offspring could be, like what different ways could these gametes combine? What offspring could they create? You see down here at the end. And then the last step is to summarize the results. So if you look at it, say tall, short. So 50% are tall, 50% are short. But when we talk about genotypes, we're going to be using just the letters. There is a practice problem for monohybrid, like it's showing a cross, about tongue rolling. And it's always helpful if you kind of make a key and you say, tongue rolling is dominant. So it would be like can roll. If you have a big T and a little t, you can roll. And then a little t and a little t can't roll tongue. So when you're identifying, okay, who's Bob, and Bob can roll his tongue and is heterozygous, you may need to flip back and use your vocab. But this would be Bob. So we put Bob up here. He'd have a T. Okay, and then his other allele would be a little t because he's heterozygous. And then Sally can't roll her tongue. So this has got to be Sally, a little t and a little t. So solve that Punnett square and analyze the results. And you also may want to try a test cross.